Okay. So looking at leaf tissue, that's where we left off. This idea that you have this cuticle, this waxy substance that is laid down on the surface of the leaves. It's actually the whole plant body is pretty well coated, including the root tissue. Root tissue is also coated with its own version of wax, not the same composition. But when you think about it, it does make sense that they kind of make most of their body pretty watertight so that they're not losing water out of cracks and crevices of their plant body, and that they have really good control on any water loss that is happening. And we talked about how that's happening with the stomata, where they can control the guard cell movement and either close over the pore or reveal the pore. And then we talked about the palisade, parenchyma cells, mesophyll, spongy mesophyll, or spongy parenchyma cells. And then we have some vascular tissue that we'll be talking about as well. And you can see one thing to cue in on is that you do have vascular tissue clustered together. It's not like you just have a single strand of vascular tissue. You have bundles of it. And so we'll come back to that idea. This is just a nice image showing you a fairly thick cuticle, like green that you see on the surface. That's the cuticle. And so just trying to give you a nice visual, excuse me, of how well covered the leaves are in this case. Parenchyma cells, remember that's the #1 type of cell in the body of the plant is the parenchyma. There's other types of ground tissue, but parenchyma wins in sheer numbers. Parenchyma cells, that's what you're looking at here. They're nothing really special to look at, but they tend to be kind of a little, they say they're many-sided. They're not like this perfect sphere of a cell. And what that does is allows for a little bit of space in here, but then you have the middle lamella kind of making sure that all those cells are staying together and staying coherent as a tissue. Here is a mock-up of stomata, and they're trying to get across the point that you have a couple of different designs of guard cells. Here on the left, figure A, you can see that the guard cells, we say they're kidney bean shaped. They look like a kidney bean. And then the ones on the right in figure B, we call those the dumbbell shape. It kind of looks like a dumbbell that you use when you're lifting weight. But no matter what the overall design of the guard cells or shape, they function the same way. And what they're trying to show here is that if you have water rushing in to the guard cells, it allows for the pore to be revealed, which is the exact opposite that I would have thought when I was first learning this way back. So what you have to appreciate is that these guard cells, if you peer into their cell wall area, they have their microfibrils laid down in such a way that it's a radial pattern. Basically, the end result is that when water rushes into those guard cells, it puts pressure on the cell wall area ultimately. And the way the microfibrils are arranged, it causes, if my hands are the guard cells, and they're closed over the pore right now in this position, but when water rushes into the guard cells, the microfibrils actually cause the guard cells to kind of move away from each other or bend or pucker away from each other. And then that reveals the pore. And so it's this water pressure pressing on the special design of microfibrils that causes the cells to move away, the guard cells to move away from each other. And then they can be full 100% open or just partly open. And they control that, how far should I reveal the pore? That's all controlled by just water coming in to the guard cells, water coming out of the guard cells. And so it's a pretty simple mechanism in a way. It's not hugely energy intensive in the sense of they don't have to put in a lot of ATP energy to get this to happen. And we'll talk more about guard cell movement later on, but basically, well, actually, when we get into water, we need to think about this idea of, well, how do you get water into a cell? Like, you got to get it to happen. And the way that plant is doing this, they create a situation where there's a bunch of solutes in the cell. In this case, they're showing potassium. So if you have, in this case, potassium coming into the guard cell, but then by the laws of osmosis, water will also go into the guard cells. And then that causes that trigger pressure on the cell wall area and causes the guard cells to open up. And then the reverse happens if the plant's like, okay, I need to close down my stomata. What they do is they just exit out. They're like, okay, so those, in this case, potassium needs to get out. And then again, by the laws of osmosis, water will follow, move out of the guard cells. And so it's really the laws of osmosis they're playing with and making sure that they can fine tune how much of that pore should they reveal. They're fun to look at under the microscope. If you take a peel off the surface layer of a leaf, that's what you're looking at here. And you can see how most of the leaves cells, they kind of look like bricks and brick walls. And then you have these unusual looking cells scattered. Those are the guard cells. And so in this case, you can see the guard cells do look kidney bean shaped. And at least some of the pore is revealed on some of these, but it looks like most of them are closed. So maybe this plant was a little stressed with water loss. And so it closed over its pore. Here, this one is a little different. So here you have brick, kind of like a brick wall design of the epidermal cells. Here it looks more like a jigsaw puzzle. So it just depends on the species, how the epidermal cells look under the microscope. But again, you can very clearly see the kidney shape being guard cells. And then this one, it looks like most of the stomata are open. You see the pore. So this plant, whoever was working with this plant must have watered it pretty well before they took the sample. So we'll come back to guard cells when we start thinking about water movement through the plant and how it works. We have to really think about guard cells in stomata in general. So let me finish up though some of the other tissue that we need to think about. Another type of tissue is parenchyma. And it really is a supporting type tissue. It helps give physical structure and support. So parenchyma, if you look at those cells under the microscope, they tend to be narrow and long. And they are living cells. They have a primary cell wall, but not a secondary cell wall. But their primary cell wall is pretty thickened up to provide that extra thickening. Probably the best example I can give you of parenchyma tissue that you've experienced is if you've eaten a stalk of celery. And when you bite through celery, sometimes you leave like the strings are left. That's a bunch of parenchyma. It's a cluster of parenchyma cells that you didn't bite through, toughly enough. And they're left kind of that string on the stalk. So the ridging on the celery stalk too, if you've ever looked at a celery stalk, it has like ridges. Those are clusters of parenchyma cells. So it really helps, in this case, it helps the celery plant have a nice, thick, strong stalk. Another type of supporting tissue is sclerenchyma. Sclerides and fibers are two subcategories. Scleride tissue can be found in a bunch of different regions of the plant body. You probably have most experience with sclerides if you've eaten pears, the fruit, pear fruit, and you bite into the flesh of the pear and it feels gritty when you're chewing it. You're eating a bunch of sclerides. Those are scleride cells that you eat, as well as other cells, but that grittiness, those are scleroid cells. And but they can be found in non-fruit tissue. It's not just pears. Fibers, on the other hand, are more, they tend to be neovascular tissue and they seem to have a pretty narrow function in the plant, in the sense that they tend to surround vascular tissue. So vascular tissue is the mechanism the plant is using to do long distance transport. It's analogous to our blood vessel system. So it's pretty important for the plant. So it does make sense that they would try to protect the vascular tissue in some manner. And so having these fibers, they cluster around the vascular tissue and give a nice physical protection. And so it's harder for things to bite through it or get poked through. The fibers are there to really protect the vascular tissue. So the sclerizing fibers, they have thick secondary cell walls. So they're really beefed up. You can notice it under the microscope. So remember, if they have secondary cell walls, are those cells alive or are they dead? They're dead, but still super important for the plant. So again, just because they're dead doesn't mean they're useless. It has important functions. Here, they're just trying to show sclerides. Parenchyma, here's parenchyma. So if you imagine the celery stalk idea, they're long and skinny. And when you look at them in cross section under the microscope, they have some interesting angles. Sclerides can look really funky under the microscope. So here you're seeing lots of that very, very thick secondary cell wall. It's really beefed up. And so all of these cells are clustered and they just have really big, thick secondary cell walls. And then fibers tend to be long and skinny. And you can see under the microscope, they're not quite as beefed up as the sclerides, but they're substantial. So these fibers, they're long and skinny, and they tend to just kind of wrap around the vascular tissue or flank the vascular tissue, totally surround it. So with the vascular tissue, now we're thinking about the plant has to have a way to move substances through its body. And, all the way from a tiny little strawberry plant to a huge giant sequoia or redwood, you have to have movement through the plant body of different chemicals. And so with xylem, that's the type of vascular tissue that is devoted to moving water and minerals that are dissolved in the water. And when you look at xylem and you look at the plant, there's a couple of different types of xylem. There's trachid, which we say are more primitive because they show up in the more primitive vascular plants. So most of you probably are most familiar with angiosperms, the flowering plants. That's usually what you get taught in K through 12. They really zoom in on angiosperms. But please don't forget, there's a lot of other plants out there besides angiosperms. And so some more primitive, so the gymnosperms, for example, the conifer trees, they are considered more primitive than the angiosperms. They only have tracheids. Whereas angiosperms, the flowering plants, they're more advanced. They have not only tracheids, but the angiosperms also have another type called vessel elements, tracheids and vessels. All right. And so angiosperms have both. Vessels, when you look at them and compare them to tracheids, they're definitely shorter, but they're wider in diameter. And that actually allows for more efficient water flow through the plant body. So the angiosperms, when they evolved, they kind of put a spin on xylem tissue and they said, let's go a little wider. And they sacrificed a little in height, but they went wider. And so when you look at an angiosperm under the microscope and you zoom into their xylem tissue, they will actually have a mixture of tracheids and vessels clustered right near each other. And I think I have a picture I can show you. So xylem tissue, whether it's tracheids or vessels, you have thick secondary cell walls. So that means those cells are dead. They're empty, no cytoplasm, but that's good because water movement can really just cook through there really nicely, quickly, efficiently. And so if you're a giant redwood, that's a good thing because that allows for the water movement to go through the whole body pretty nicely. The other type of vascular tissue is called phloem. And xylem and phloem, when you look at a plant under the microscope, those cell types will be right next to each other. They're clustered. I'll show you a picture. Phloem is not for water transport per se. They're more for doing sugar transport, which is dissolved in water, but it's really that's their mechanism. They'll do photosynthesis, they'll make all this lovely sugar, and then they'll dump it into the phloem tissue and send it on its way to other parts of the plant body. So again, you can imagine if you're a plant doing photosynthesis, that's in your leaf tissue, which is, yeah, okay, great, but other parts of your body need that nourishment. And so, especially roots that can't do any photosynthesis, so you really have to have a nice way to transport that sugar. So phloem is very good. Now, phloem is a little different than the xylem because they're actually living cells. So that's an added little complication. But when you look at phloem with different plant species, the more primitive type are the sieve cells. So gymnosperms are the more primitive type. So they have a version of phloem called sieve cells, whereas the angiosperms that are more advanced, they did their own little version of phloem, and they came up with sieve tube elements, which I often abbreviate down to STE. And then TC is another thing that they evolved over time. Those are called companion cells. Companion cells. So sieve tube elements and companion cells, when the angiosperm is building those, they'll build a sieve tube element and then they'll put a companion cell right next to it. And then a sieve tube element and then a companion cell right next to it. And they named it companion because those cells are a companion to its respective sieve tube element. They're like 1-to-1 ratio. I'll show you a picture. And so sieve tube elements and companion cells, it was a good design that they evolved over time because it makes them more efficient than just the sieve cells that we see in gymnosperms. So again, the angiosperms really played around through the process of natural selection and selection forces. They were able to create these, vessel elements, sieve tube elements that just made them more efficient than the more primitive plants. And so that's one reason why angiosperms kind of dominate our planet. They're the largest group of plants, and this is one reason for that. So here under the microscope, or if you were to look under the microscope, you might see something like this. So xylem is on the left, and you can see they're trying to show that there's some interesting cell wall. They kind of ramp up what's going on in the cell wall to really make it more rigid and structurally sound. And then tracheids are on the left. You can see how tracheids are they're longer, a little skinnier. And then the vessel elements here are a little bit wider, especially this one on the far right of the vessel element example, you can see. And then we'll come back to this idea. This is a weird, they call it a sieve plate. They're just special designs at the end of each cell. So what you have to imagine is that these tracheids or the vessel elements, what they do is they stack on each other. And they just stack end to end to end until they form this very long vascular tissue that goes through the whole plant body, all the way from the roots all the way up to the leaf veins. It's all continuous. And so it's a pretty massive undertaking. So when you have these two cells meet end to end like that with the vessel elements, you have these special, what we call sieve plates. We'll talk more about that. Phloem here on the right. So they're showing sieve cell here, long and skinny. To be honest, they don't know a ton about sieve cells. There hasn't been a ton of research with sieve cells, and so it's still rather mysterious. They know way more about sieve tube elements because that's in angiosperms have sieve tube elements that are, actually angiosperms have both. Well, I'm sorry, they have sieve tube elements. They know more about sieve tube elements and companion cells than they know about sieve cells. And so they're trying to show here that it's a one-to-one relation for every sieve element. They have funky granite walls too, sieve plates. So we'll talk more about those. Here's another depiction trying to show that you have a sieve tube. Some people call it a sieve tube member. I tend to sieve tube element with an associated companion cell. So sieve tube elements are living. That means they have a primary cell wall, no secondary cell wall, but they also have cytoplasm. Now, when you look at it more carefully, they'll state sieve tube elements and sieve cells are living, but highly modified. What they mean is when they look in the cell, in the sieve tube element, yes, they have cytoplasm, they have some organelles, but not all. Also, they've done experiments. If you take its associated companion cell and you put in a poison to kill that cell, the sieve tube member it's associated with dies. So that's why they needed a companion cell. They know that the companion cells are supporting each sieve tube member or element physiologically because the companion cells are a fully functioning cell. And so I think this was, they hypothesized that this is allowing the sieve tube element to have less stuff in the way of movement. You know, so as the sugars are coursing through the sieve tube element, they had all the organelles in here. That wouldn't be terribly efficient, right? The sugar solution would be bumping into everything. It'd take a heck of a lot longer for the sugar to get where it needs to go. So some of the organelles are missing in the sieve tube element, and the companion cell has to make sure it stays alive, but makes enough energy to support the sieve tube element. Yep. Yep. It would be. It would be more efficient if it was empty. And why didn't it do that? Good question. I don't know. But yeah, I mean, because Xylem does it. They figured it out with Xylem. But yeah, I think yeah, that from what I've read, there's been some hypotheses out there, and could plants eventually, angiosperms eventually evolve to be empty? Maybe, but certainly not in our lifetime. So yeah, it's an interesting question, and I don't have a solid answer. There's some hypotheses out there because sieve tube elements do more than just sugar movement, and so, but I don't know. I don't know enough about it to really weigh in too heavily on that. But sieve tube elements, you know, they do more. They do know more about them because that's what shows up in the angiosperms. They don't know very much about sieve that show up in the gymnosperms. So here's a cross section of part of the plant body. And so one of the things on here, you're actually looking at root tissue. And the reason I can say that with full confidence, these things that are coming out here, eventually going out really long, these are actually lateral roots that are starting to form and grow out of a more central root. So that's how I can tell it's a root. Also, I can tell it's a root based on how the vascular tissue is arranged, but if you take in Plant Kingdom, we get into that quite a bit. We won't do that in this class, but this ring of cells here that looks very distinct in the sea of all these other cells, right? And so all these other cells are parenchyma cells, right? So these kind of bluish green areas that you see all through here, that's all parenchyma cells, right? And so in root tissue, parenchyma cells are really good at storing starch. So right now, we're almost into September. So right now, some of these, especially some of the trees, are starting the process of getting useful stuff from their leaves and getting it down into their roots to store up over winter. And a lot of the times they'll convert that into starch for long-term storage. So all that green is parenchyma cells. Then that ring of tissue that you see, that's vascular tissue. So the couple things I want you to grasp from this image is that when you look at the vascular tissue, it's clustered. So these big open cells, by the way, all of those big open ones, those are vessels. So you can see how much bigger in diameter compared to all those little cells that you see here, the clusters of smaller cells. So those are tracheids. These are vessels. Big difference in their species between the two. And then there's some fibers down here there's fibers up here. Okay, so there's some fibers. It's a little hard, grainy, out of focus, but fibers tend to form a circle. Circle of wagons around, it's sort of getting this circular protection around the xylem. And here again, up here, there's some fibers all the way around. So trachea, vessel, and then the phloem tissue is here too. It's a little hard to see, but maybe you can you see that it's kind of a bluish green. You see kind of up here, clustered here. I don't know if you can pick that out from where you're sitting. But that's sieve tube elements that are clustered together and companion cells. So one of the big ideas to think about as you're thinking about movement through the plant body is that you have this clusters of vascular tissue, clusters of sieve tube elements, clusters of vessels and tracheids. And that's good. They're not putting all their eggs in one basket, so to speak. So, if one of the sieve tube elements, if one of those fail, they'll have a neighbor, hopefully, that's good to go, which I forgot to point out in this slide. You see all these pores here? So those are pores. So when you have all these cells stacked, clustered right next to each other, you have to imagine, for example, another sieve tube element smacked up against it, and they can pass nutrients between each other or substances between each other. So if one sieve tube element highway system fails, hopefully the neighbor, they could, redirect and say, here, take it. I've been damaged somehow. Maybe a bunny rabbit came along and bit into it, but they have the ability, because they're all clustered like that, to move the nutrients to hopefully a fully functioning xylem or phloem. Here's another root from a different type of angiosperm. And so you can see their vascular tissue is very differently arranged compared to what we just looked at. Here, this is if you're curious, this is a monocot root. This is a dicot root. Here's the vascular snap in the middle of the root. And the xylem is the big red cells. And then the phloem, in this case, let me see if I can circle. The phloem ends up being like in the crevices of the I often say that the crevices. But again, the xylem and phloem are all clustered. And there's many, many of them all clustered together. And in this image, you can see all the purpley stuff in the parenchyma cells. Those are starch grains. So whoever prepped the slide stained it for starch. And so they must have used iodine when you put iodine on starch, it turns purple. Here is a stem tissue in cross section. And again, the vascular tissue. If you look at it, it kind of forms a ring all the way around, but there's like these discrete bumps, but it forms a ring all the way around. But again, the xylem has stained red. They usually will stain it so it picks up the secondary cell wall. It's reddish purple. And vessels are the bigger ones, tracheids are the smaller ones, and then the kind of bluish green cells are the phloem. You can see that the xylem cells, really thick secondary cell walls. Phloem, no, they have primary cell walls, they're living, they're more delicate looking. So here again, vessel traces are in here, tissue is highlighted. So we're going to be talking a lot about vascular tissue in this class because we need to think about movement of nutrients, water through the plant body. So take a look at those images and more as you're studying to get this, and then it's analogous to the blood vessel system. If you think about that, I think that does help. Are there any questions on just kind of the basics of plant anatomy? Any portion of today or any of the other stuff we've been talking about so far? Okay. Well, we're going to start zooming into xylem. All right, so the xylem is where we're going to see all the water movement and the minerals that are dissolved in it. So we need to think about water in a little bit more detail. And there's some interesting properties about water that makes it very unique. I think your slide set that's posted in EDUCAT has a few more slides than what I'm going to cover. So make sure that you are looking at the posting in EDUCAT. I'm going to work through this pretty quickly. My assumption is you've had information on water in basic biology and chemistry classes. I'm just going to hit some of the highlights to get you thinking. So here, certainly we have H2O. You can see there's partial positive and negative charges, which give water very unique properties, where you have the bonding, and you have to kind of imagine the water molecules constantly binding and breaking their bonds with each other. As you are thinking about, a glass of water, for example. And when you have the idea of, you have partial negative and positive charges on this water molecule, it sets up some really interesting situations. Hydrogen bonding, of course, between the water molecules. And when you think about water, like in the xylem, for example, or a glass of water, you have this idea of water movement continually going. You have bonds breaking and forming continually. When you think about water in the plant body, we need to also think about the xylem tissue because the water is going to be in physical contact with that as it's moving. So we need to think about water interfacing with, in this case, xylem. So we'll be talking about that too. Here are some properties that you should have learned in more, basic chemistry or biology. When you think about specific heat of water, you're looking at the amount of energy that it can absorb to get it to rise, 1 degree Celsius, for example. And water has the highest specific heat out there, I believe. Thermal conductivity, that's the idea. I like the analogy if you, heat up a fire poker into the fire where it gets red hot, and then you put the poker into a barrel of water, for example. And it's like, okay, how long does it take for that hot to radiate into that barrel of water, for example? And so basically we're thinking about the idea that water is just really good at conducting heat away from the point of application. And so I like that imagery of a fire poker getting doused into a big barrel of water. You imagine yourself having a fire in a fire pit, but you have your barrel of water there for safety. All right, so thermal conductivity, water is really good at being able to disperse that heat away from the point of application. And heat of vaporization is another thing to think about. This is the idea of, how much energy does it take to separate molecules out of the liquid phase and into the gaseous phase. And we need to think about that a little bit when we get into transpiration, which is a fancy word for water moving through the plant and ultimately out through the stomata. That's the process of transpiration. So we'll be talking about that after we learn a little bit more about water and water potential. Okay, so this is all posted, so you don't have to write all this down. The other thing too about water is it has this incredible cohesive property where their water molecules are very attracted to each other by their nature of how they're configured. And that gives rise to nice surface tension on the surface of a body of water, whether it be, water in a beaker, or I like to use the analogy of a pond. We have lots of local ponds and lakes around here. And I love watching the water insects, as they walk across the water. You're like, how can they do that and not sink? And it's because on that lake that's allowing for them to walk across the water and not sink. And so what you're thinking about here is on the lake surface, you have those water molecules that are on the very, very surface of the lake. They're interacting with the water below them, but they're also interacting with the air above them. And when you have that interface like that between water and air, what actually tends to happen is those water molecules tend to be more attracted to the water below them than the air above them. And so it creates this tension. They often refer to the surface water as kind of like, it behaves like an elastic membrane. And it gives some good tensile strength to that layer of water. And that's what's really allowing those water bugs to just walk across the water and not sink. It's because there's that surface tension going on. And so when you think about tensile strength or surface tension, you can measure, it's kind of weird to think about measuring tensile strength of a liquid. They do it like with steel beams and things like that to make sure they're strong enough for a skyscraper. But you can do it with liquids. And really, that's a nice analogy where you put it into a plunger like that and tap the end where the needle goes. And then if you pull back on the plunger, you're putting tension on that column of water. And the definition of tensile strength is what is the maximum tension force that you can put on that column of water before it breaks, in the sense of, in this case, it would be gases would come out of the water and there would be like a air gap essentially in between the water molecules. And so, which reminds me, that's a really good thing to think about when you think about a plant and water movement. It's not pure water. It's got gases in it, it's got minerals in it. And so they're grabbing that water from the soil environment. It's certainly not pure water. All right. And so we need to think about that idea as we think about how does the water move through the plant body and how efficiently does that happen. So cohesion is the idea that water molecules are really attracted to each other. And certainly at the interface, the very top layer of water is much more attracted to the water below it than the air above it. The other major idea is adhesion, which is the idea that you have water attracted to some solid surface. So for what we care about in this class, for right now, we're thinking about the xylem. All right, so how attracted is water molecules to the xylem? It's moving along inside the xylem cells as it moves through the plant body. And so we need to think about that too. And as we get more into transpiration, we'll talk a little bit more about that whole system. All right, but before we get into the actual process of transpiration, I need to talk to you a little bit more about water potential. Does that phrase sound familiar? Water potential? Maybe. I generally seem, when I ask students, it seems most students don't really know much about water potential. It doesn't get taught very much in other classes. So when we think about water potential, what we're really trying to grapple with is how does water move from point A to point B in different contexts or situations. And one aspect we need to think about is diffusion. And you've learned about diffusion. I'm pretty confident all of us here have the basics of diffusion down. It's actually proposed, the law of diffusion was proposed by Dr. Fick, so they named it Fick's first law of diffusion. And it is defined, so J, the equation that you see up there, that's the equation for the rate of diffusion. So J is the symbol that they use for the rate of movement. And then the D is your diffusion coefficient that you can look up. That's provided to you. And then the delta, dC over dx, or delta, the Greek delta, delta C over delta X is how I learned it, but some people just put dC over dx. That's your concentration gradient. Oh, what happened there? Huh. Okay, so that should be shifted down. Well, yeah, it's okay. dC over dx is the concentration gradient. I guess that's good enough. All right, so what that equation is basically saying is that when you think about diffusion, you're thinking about a concentration gradient. dC over dx is a concentration gradient. So then we say, well, you know, how steep is that gradient? How shallow is that gradient? And that will impact how quickly things will move. So usually when they teach diffusion, they often say something like, you know, you drop a sugar cube into a cup of coffee and you watch it diffuse into the coffee, or you open a bottle of perfume in an air in a room. How long do the perfume molecules take to diffuse out of that very concentrated high perfume solution into the dilute air? And so this idea of diffusion, when you look at delta C over delta X, we'll come back to that idea. We also are thinking about with diffusion properties, when you look at the plant body and you think about diffusion, it works great over really small distance in the plant body. So like if something has to move just a really short distance, diffusion is adequate. But when they did the calculations and said, I wonder if water's moving through the plant by diffusion, ooh, no, it would take like hundreds of years for water to move even through like the height of a corn plant. And so they quickly figured out, oh, water's not moving through the process of diffusion, at least long distance. It's effective for like within the cell or maybe two neighbor cells. Yeah, diffusion. But through the plant body, no, it takes way too long. Right? So there's another strategy the plant is employing. But we got to save that for next week. But you all know there's no classes. So I will see you on Wednesday, I guess. And then right after that, we have lab where we're going to do an experiment on water potential in lab. So I'll try to get you what you need on Wednesday to get you prepped for the lab.